Now I'm going to demonstrate several different things you can do under the title Enzyme Structure and Function. The other thing I'd like to point out before I start is this shows you quite clearly the versatility and the flexibility of the model. Just as in the other two kits that I've produced, the RNA DNA protein kit and the cell membrane protein kit, you can do several lessons with the same pieces. And in fact, you're going to see here as we progress through this particular set of lessons where you can actually get ideas to create some lessons that maybe I haven't even thought of with the same pieces. And that's that layering power of the model. So the reason why there's an arrow here is to show you power magnet here, and with this polypeptide chain that was involved in the fourth level protein folding, I'm going to break the bond right here. So now we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm going to take those 10 pieces here, and I'm going to fold those up into this kind of shape. It's like a mini fish, and that is going to be our model of a competitive inhibitor. The larger purple section that you're left with, I can still do that same kind of what we call fish move, right? So see, that's kind of like a fish now, but it's larger, and that's our substrate in this particular lesson. So quite clearly you can start to see how you can multitask, so to speak. This arrow is to show you 2, 4, 6, 8, 10. I'll break that bond between these two power magnets. And I'm going to make this what we call like a C type of structure. And that's hanging like that, and that's totally fine. This is going to be our non-competitive inhibitor in this particular lesson. Now we're going to take the remaining part of the multicolored polypeptide chain and we're going to fold it into a three-dimensional protein. And again, I don't have any particular real shape in my mind right now, and even when I teach this, it's something that I don't have to worry about because it just starts to kind of happen when you start to play with it. One thing that I am thinking of though is we're going to do the induced fit model. So I know I got to build a top and a bottom, so I'm thinking of that. Right? And I'm thinking, okay, I got to create an allosteric site and an active site, and I kind of want to make it globular, so I give it that kind of structure here and here, and you know what? That's not too bad, right? So what I'd like you to see that's in my brain here, here's the allosteric site in this particular example, right here, this opening, and the active site is right here. So, with the active site, I'm going to demonstrate with the substrate. The first thing I'm going to show you is the induced fit model, and we're going to now talk about before, during, and after diagrams. This is a classic situation where the students can follow quite clearly those three types of diagrams. Okay, I'll leave that there for now. So the active site can come down. So I would show the students, okay, I tell them I want you to make a before diagram. They have sketched this enzyme. They know where its active site is. And I'm bringing the substrate closer to the enzyme. And really, that is what I would like them to sketch for the before diagram. Just really, the way I teach this is, if we could stop the reaction just before it happens, what would the picture look like? So the students take that picture or put the picture on their paper and they'd make some written notes. The during reaction, again, the students now draw the substrate in the active site, and with arrows they're going to show, and the great thing about this particular protein is you can start to manipulate it. And sometimes I'll do this, and look, here's a couple things that happen on the fly, which is fine. That little Velcro came off. That's no big deal. But you could, to demonstrate the fact that the protein is breaking the bond, see what I'm doing? And boom. So I'm showing that the protein is breaking those bonds. And as you know, before that happens, right, this enzyme, and here's the induced fit mechanism, the enzyme collapses around that substrate. The students are showing two things with arrows. The enzyme breaking the bond, and with arrows they're showing the enzyme changing shape. Okay? That's their during diagram. So now that substrate is no longer in its form, so I take that off the board. The during diagram is over. So the after diagram, the enzyme is going to open up like so, go back to its original shape, and now we have two smaller molecules, again, whatever they might be. And in this particular, it's a very generic lesson, so the students might ask, what are the names of these? And feel free to ad-lib whatever you'd like. So there's our two products. Now, to demonstrate a competitive inhibitor, I have the mini fish. Remember, this large polypeptide purple was a fish, and the competitive inhibitor is just simply a mini fish, and I just communicate to the students that if this particular molecule is in the active site, the larger substrate can't fit in. So this has shape specificity enough to bond into the enzyme and to block that substrate from entering. For the non-competitive inhibitor, this kind of what we call a C move, we just bring this over. I would take the non-competitive, or the competitive inhibitor, I'm sorry, I take the competitive inhibitor out, and now I'm modeling non-competitive inhibitor. So it's going to come over, and see how I can squeeze it right in here to the allosteric site. And what I'm trying to communicate to the students is that chemistry changes the shape of the protein and it closes. And that is very cool. And now they can see the active site is not capable of receiving a substrate. So that covers the competitive and non-competitive inhibitors. The other thing I'd like you to try to focus on is in the kit, you also were given 2 glucose molecules, and you know that those 2 glucose molecules make maltose. So if you get into a more specific, say, digestion lesson, that's why we put the 2 glucoses in, you could make the enzyme, right? So the enzyme that's going to act on maltose, as you know, is maltase. So again, be creative. You can create maltase and break that bond if you're teaching digestion. And 2 things for lactose. So that's why we put 3 glucoses and 1 galactose in. 2 glucoses give you maltose, glucose and galactose give you lactose. Again, during digestion, you can design a different enzyme to break lactose apart. And the other thing that's really cool, again, with the 3 kits that I've developed, if you buy or had purchased the RNA, DNA protein kit, there's a lac operon lesson in there. And if you had this protein folding kit, you could really take that to another level in terms of showing 3-dimensional proteins. Now, the last thing I'd like to show you that's going to end all these lessons that are outlined on your package is how simple it is to show denaturing. So we, again, come back to this enzyme that I've created, and however you want to talk about denaturing with heat or pH or whatever, you can just clearly see that I'm going to change the shape of the enzyme and see how I can get that to hang down. And those power magnets are strong. So I'll hang it over here, attach that back, and maybe take this part, hang it down, and it's kind of like there, right? It's limp, it's dead, it's non-functioning. And that's really kind of the structure we show our students to say that this protein is now bent out of shape, and that demonstrates clearly denaturing. So that concludes my demonstration on some protein enzyme structure function lessons.